Hello everybody, I'm Tyler Schrader and I will be discussing Marr's lab where we do microrobotics research. We have 3 postdocs, Matt, Eric, and Ramona. We have 4 graduate students, Dante, Yan, Coy, and Aaron. We have 1 master's student, Brandon, and 2 undergrads, myself and Logan. Next up, we're going to be talking about how we actually get these micro robots to move. Here we've got 2 dimers rotating under different applied magnetic fields. As you can see, their motion is quite different, and we're able to control their motion based on varying the magnetic field of the system. Next up, we're going to be talking about one of the applications of our research where we use microwheels to break up blood clots. We functionalize these wheels with the enzyme TPA, and we are able to both mechanically and enzymatically remove our blood clots. So here is a video of that. Here at the top, we have our Microwheels with the applied corkscrew magnetic field. And below that, we have directly spinning microwheels. And then we have just soluble TPA in our other 2 frames. As you can see, we are getting increased lysis rate with the functionalized microwheels with the corkscrew pattern by quite a large margin. Now that we have the preliminary data from that experiment, we've been transitioning into more complicated models, such as the in vivo model, where we use 2-photon microscopy to analyze the system in rats, and then we use microfluidics to model potential vein systems. And here is a quick video of one of our microfluidic systems. The other primary application of our microwheels is in the treatment of lung-based illnesses, primarily cystic fibrosis, where mucus fills the lung cavities. So we aerosolize our wheels, and then we can deposit them onto our mucus and then functionalize them with papain, and we can potentially remove mucus and help the body naturally export the mucus. So here we have our aerosol characterization, where we are measuring both aerosol radii and microwheels movements based on whether or not they were aerosolized or just placed into our system. We use oil emulsion to characterize our aerosols, and that is the project that I currently work on. So here is some interesting videos relating to that characterization. Next up, we've got a quick video showing in real time droplets right after being aerosolized, And as you can see, they can combine very quickly. Here is a video showing a racked focus of a droplet with microwheels suspended inside. And as you can see, these microwheels have been sitting for quite a while, so they've settled at the bottom, and then a few still remain suspended in the droplet. Next up, we've got a video of microwheels being created from a sprayed bead collection. And as you can see, they combine to create larger wheels and very interesting structures. And in this case, we were having some issues with some microwheels sticking to the surface, as you can see. And then we've been also working with mucus synthesis and characterizing the viscosity of our mucus and how that changes with our microwheels and papping system. Next up, we've got our magnetic chains where we chemically bond our microbeads to one another in a linear fashion in order to create novel structures and movement. So here we can see flagella-like movement where we have a large bead attached to a chain of smaller beads, and we can induce that rotation. And then we can also induce a tail whipping rotation and arch stretching. And to get a better idea of what that is, here is a video. Last up, we've got our microspheres, which are spheres about an order of magnitude bigger than our microwheel spheres that are chitosan based and then loaded with iron-based particles so that they can be controlled using a magnetic field in a similar way. And their scale with relation to epithelium topology. And here in the corner is a fluorescent image of the actual spheres. That's the end of our presentation. If you're seeing this before the 23rd, I hope to see you at our poster session. If not, I hope you're able to reach out, and hopefully we'll see a few of you joining the team. Thanks.